everyone. Today I'm going to be talking about the regulation of the Lac Operon. Before we dive in, we need to talk about some terminology and functions of operons. First things first, what is an operon? An operon is a group of structural genes and regulatory regions of DNA that control their expression. Structural genes encode proteins that are used in metabolism or have an important structural role in the cell. Regulatory regions can be genes or elements that affect the transcription and the translation of DNA. One benefit of operons is that they allow multiple genes to be made at the same time or to not be made at the same time. The genes of operons are transcribed into polycistronic mRNA. This may be a familiar term to you, but to review, polycistronic mRNA encodes multiple genes on one strand. In terms of the Lac Operon, 3 gene products are produced from the 3 structural genes that make up the operon. To power and help regulate the Lac Operon, lactose is broken down into galactose and glucose and used as an energy source. Some of the environmental lactose is broken down into allolactose, which is a key regulatory element of the Lac Operon, which we will talk about in the next few slides. There are a few components of the Lac Operon that are important for its regulation. The regulator gene, LAC-I, produces a repressor molecule that prevents transcription from occurring by blocking the LAC-P site. The promoter, LAC-P, is where RNA polymerase binds for transcription. The operator, LAC-O, is the site where the repressor binds to block transcription. Then we have the 3 structural genes of the Lac Operon. LAC-Z, which encodes for beta-galactosidase, LAC-Y, which codes for permease, and LAC-A, which codes for transacetylase. Under normal conditions, the genotype for the Lac Operon is written as I+, P+, O+, Z+, Y+, A+. For this scenario, I+ means that LAC-I will produce a functional repressor. The presence of lactose means that there will also be allolactose present. The allolactose molecule binds to the repressor molecule, which inactivates it. As you can see in the picture, with allolactose bound to the repressor, it will not be able to fit into the LAC-O site. This means that RNA polymerase will be free to bind to LAC-P. Because the repressor molecule cannot bind to LAC-O, RNA polymerase is free to bind to the promoter region, LAC-P. RNA polymerase carries out transcription of the Lac Operon, here transcribing LAC-Z, LAC-Y, and LAC-A. Now we have the same genotype of the Lac Operon, but instead in the absence of lactose. Just as before, LAC-I produces the regulatory protein, the repressor. However, in this case, allolactose is not present and is not available to bind to the repressor molecule. Because of the absence of lactose, and therefore the absence of allolactose, the repressor is free to bind to the operator region, LAC-O. This also blocks the binding site on LAC-P for RNA polymerase. As you can see here, RNA polymerase is blocked from accessing the promoter region, LAC-P. Because RNA polymerase cannot bind, there is no transcription and none of the structural genes are produced. Now that we have gone through the normal functional genotype of the Lac Operon in the presence and absence of lactose, we must cover some of the mutations the elements can have. The regulatory gene, LAC-I, comes in 3 forms. I+ produces a functional repressor. I- means the produced repressor has a bad binding site and cannot bind to the operator. And IS stands for a superrepressor, which means the repressor always binds to the operator and never binds allolactose, even when lactose is present. The operator can have a mutation denoted here as OC, meaning the operator is constitutive. This means the operator cannot bind a repressor even when allolactose is not present and the produced repressor is available. The promoter can have a mutation, P-, which means the promoter cannot bind RNA polymerase. This means no transcription would follow on the operon. The structural genes can also have mutations, Z minus, Y minus, and A minus, which result in non-functional proteins. Now that we know all about mutations of the Lac operon, let's go through some examples. For this mutation, we have a partial diploid, hence 2 copies of each and 2 operons pictured. This is a common way you will see mutations of the Lac operon presented. Here we have I minus, P plus, OC, Z plus, Y minus, A plus, and I plus, P plus, O plus, Z minus, Y plus, A plus. Contrary to what you may think, both operons here will produce a functional repressor. This is because the I plus of the second genotype is transdominant to the I minus of the first genotype. Whenever one copy of I plus is present, functional repressors will be produced for both copies. Allolactose is also produced here as a result of the presence of lactose. This means that, in both cases, allolactose binds to the repressor and inactivates it. Next, both genotypes have P plus alleles, so both LAC-P regions are open for binding by RNA polymerase. There is also a constitutive operator on the top operon. Here, LAC-O cannot bind the repressor molecule anyway, because the repressor was inactivated by the presence of allolactose. But it is important to remember that a constitutive operator means a repressor cannot bind even when allolactose is not present. This is only applicable to the corresponding strand because it is a cis-acting mutation, meaning it only affects the surrounding regulatory elements on its own operon. Because neither strand is repressed by the regulatory molecule, RNA polymerase is free to bind LAC-P in both scenarios and transcription proceeds. In the top image, the structural alleles are Z plus, Y minus, and A plus. This means only the beta-galactosidase and transacetylase genes produce functional proteins, while no functional permease is produced. In the bottom image, the structural alleles are Z minus, Y plus, and A plus. This means no functional beta-galactosidase is produced, while functional permease and functional transacetylase are produced. When you take both strands into account, LAC-Z, LAC-Y, and LAC-A are transcribed and create their functional protein products. And now for our second example. This is the same partial diploid mutant genotype, I minus, P plus, OC, Z plus, Y minus, A plus, and I plus, P plus, O plus, Z minus, Y plus, A plus. But now lactose is absent. As before, the repressor is produced by LAC-I on both operons because the I plus allele is transdominant to the I minus allele. It can act on both operons to overshadow the effects that I minus would have. We are also operating in the absence of lactose. Therefore, no allolactose is present to bind to the repressor molecules in either scenario. The absence of allolactose means that the repressors are free to bind to LAC-O. In the top figure, we can see the importance of the constitutive operator. As mentioned before, the constitutive operator is a cis-acting mutation, so only one of the operons is affected. The constitutive operator blocks the binding of the repressor molecule, while the normal operator allows the binding of the repressor, as you can see in the figure. Now, RNA polymerase can bind on the operon where the operator is constitutive because the repressor was unable to access the LAC-O binding site. On the operon where LAC-O was not mutated, RNA polymerase cannot bind because the repressor is blocking the binding site on LAC-P. The bottom operon has no transcription, but the top operon is transcribed. LAC-Z and LAC-A are not mutated, so functional beta-galactosidase and functional transacetylase are produced. LAC-Y is mutated here, so functional permease is not produced. None of the genes on the bottom operon are transcribed, so no functional protein products are produced from the bottom strand. Now we're going to go over different control methods of the Lac operon. First, we have the Lac operon under negative inducible control. Under negative control, the regulator molecule is a repressor. In the Lac operon, this occurs when Lac I produces a repressor molecule. When lactose is absent, the repressor molecule binds to Lac O and transcription does not occur. When lactose is present, we see the inducible control aspect. Under inducible control, transcription is turned on when the small molecule is present. In terms of the Lac operon, the small molecule is allolactose, which is only produced when lactose is present. Allolactose binds to the repressor to block the binding of the repressor to Lac O. Therefore, RNA polymerase is able to bind Lac P and transcription can occur. Now we're going to talk about the positive inducible control of the Lac operon. This includes two new elements that haven't been mentioned yet. The catabolite activator protein, also known as CAP, and cyclic adenosine monophosphate, commonly referred to as cyclic AMP. These two elements are needed to help the binding of RNA polymerase to Lac P. The CAP molecule helps the weak Lac promoter bind to RNA polymerase for transcription. Cyclic AMP functions as the small molecule and binds to CAP to ensure a strong bond between CAP and the Lac promoter. Here we have cyclic AMP and CAP complex bound to Lac P to assist in binding of RNA polymerase. Our positive control aspect is the CAP regulator molecule acting as an activator. When CAP is present and bound, transcription occurs. The inducible control aspect from cyclic AMP, which acts as the small molecule. When cyclic AMP is present, transcription is on and is increased. That's all I have for today. Thank you guys so much for watching. I hope you enjoyed and learned a lot about the regulation of the Lac operon.